Well, welcome back to the third of a series of videos on the mathematics of biology. Um, this one can be viewed independently of the first two, as we will not be relying upon any real insight into the chi-square problem. We're going to introduce here some techniques for going from phenotypes to genotypes using so-called Hardy-Weinberg analysis. Okay, so let's dive right in. So biologists have really figured out some clever ways of looking carefully at a population and noticing the phenotypes of a particular trait, say, and from that inferring both the allele frequency in that population's gene pool and ways to calculate the genotypic distribution of that same population. So whether or not something like winds up being big A, and or little a, and so on. All right, this is pretty incredible. We can really, by just looking at the population carefully, absolutely determine what's going on in the gene pool. So let's, before diving into the technique, let's just remember that we're really used to moving in this direction from genotypes into phenotypes. We can use Punnett squares with parents whose genotypes are assumed to be known, big A, little a, and so on. We can cross those and then determine, what the population should look like. Okay, what the phenotypes should look like. But what we're talking about here is something different. We're talking about looking at a population, say lots of flies, and there's 7 of them shown here, but maybe 700 of them, and from the population, figuring out what must be going on in the gene pool. So that's what we're going to try to do. All right. So hopefully you are amazed by that and are ready to jump into how it gets done. We have to make a couple of kind of definitions here. We'll do an example and we'll develop what are known as the Hardy-Weinberg relationships or the Hardy-Weinberg equations. But we need to have some definitions set. Okay. So we'll just start by thinking about a plant that has a gene for tall or short, and that it's a typical dominant recessive type of arrangement where big T is dominant over little t, and those alleles code for the height of a plant. So then we say that if you could lay out all the alleles for that trait for an entire population, if you could lay them all out and figure out the probability of how many big T's there were, and the probability then of inheriting a big T from a parent, we call that probability P. And then the other probability of inheriting the small t, lowercase t, is then Q, right? So in other words, given all of the genes in all of the plants in a given population, big T makes up P% of the alleles and little t makes up Q%. So if P was 0.75, we would say that it makes up 75% of the population. Parents can, sorry, there's a typo there. Parents can, of course, be heterozygous or one of 2 homozygous combinations of big T and little t. All right, and just to add in one more assumption that all the crossings are randomized, that there's no favored kind of crossing. It's not that the big T flies will always seek out big T fly. I'm sorry, why am I saying flies? Plants, that the matings are randomized, okay? And I should say that it's when we say that one cross is as likely as another, we're saying that it's as likely as the gene pool will allow. So in my example here, if big T for whatever reason makes up 90% of the gene pool, then there'll be a small number, a small percentage of the crosses involving little t. But so there's nothing favored here. It's just all the matings can be are random. Okay, so let's see where we go with that. Hardy and Weinberg were 2 20th century, I think 20th century biologists who came up with these ideas. They said, hey, listen, if we assume that there are these 2 alleles that govern a particular trait, and there's only 2 alleles that govern the trait, then the probability of one allele plus the probability of the second allele has to add up to 1. If this is 75%, then Q has to be 25%. Okay, so that's pretty straightforward, and I hope makes a lot of sense. But then you can start thinking about the genotypic combinations. This is a single allele, big T having a probability of P, little t having a probability of Q. But the probabilities of genotype combinations are kind of hidden within this equation here. P squared is the probability of an individual being homozygous dominant, and Q squared is the probability of an individual being homozygous recessive, and then 2PQ would be the probability of an individual in this population being a heterozygous. And we'll talk a little bit about that in a moment as we go through an example. So those are two different equations. One is about the gene pool, the allele frequencies, if you will, and the other is about how those alleles can be combined, okay? So what are we trying to do? We're going to look at a population and we're going to try to establish both P and Q. Those are what are called the allele frequencies, as we've been saying. And we'll try to determine P squared, 2PQ, and Q squared. Those are the genotype frequencies, the ways in which the alleles are combined. Of course, if we have both P and Q, then it becomes trivial to calculate the parts here in part B. So let's take a look. Here's an example. Let's say we looked at 500 plants in a population and we found that 400 of them are tall and 100 are short. So what can we do with this? Okay, here's our first step. Key first step in any kind of Hardy-Weinberg analysis, okay? Focus in on the recessive population, all right? But these 100 are going to be the key to the entire understanding of what's going on, okay? Why is that? Well, the only way to be short is if you're homozygous recessive. That's the only combination that leads to being a short plant. So the odds of inheriting T, little t, from one parent is, as we said, Q. But the odds of inheriting T from both parents is Q times Q, or Q squared. So what do we know? Right away we know that Q squared is equal to 100 over 500. Q squared is equal to 0.2, all right? So if we take the square root of both sides, square root of Q squared is Q, the square root of 0.2 is whatever that number is, okay? We take the square root of both sides and we wind up with this, that Q is equal to 0.447, okay? That is the square root of 0.2. You got to be careful there, like you're dealing with decimals, numbers that are less than 1, and it may surprise you to see Q be a larger number than Q squared, but it makes sense if you give it a few moments thought mathematically. It also must follow, since P plus Q is equal to 1, that P is equal to 0.553. Those have to add up to 1, okay? So boom, we've got it. We've got the allele frequencies. By just looking at the recessive population, we unlock what Q is and then can immediately unlock what P is. So we now know that the allele frequencies for this particular population, for this trait, well, is 55.3% big T and 44.7% little t. So that's great. We've accomplished something interesting. Well, once we know P and Q, it should be pretty straightforward to get the genotype combinations. How do alleles combine in these pairs? Well, we have our definitions from before, P squared, Q squared, and 2PQ. So we do the math. P squared is P times P, or 0.553 squared, which turns out to be 0.306. And Q squared is 0.447 squared, which is equal to 0.2, but of course we knew that already. Actually, that was the very first thing we knew. So in all honesty, when we do any of these kinds of problems, Q squared, the genotype combination, that is tied to the homozygous recessive trait, that's the first thing that we know. Okay, but anyway, it's good to confirm it here that we're back to 0.200, and then of course Those two have to add up along with the 2PQ, the heterozygous combination, right? These heterozygotes. That has to add up to 1. So 0.306 plus zero point 0.2 plus the 2PQ has to add up to 1, and indeed, that indeed happens. So if you do 2 times 0.553 times 0.447, you should get 0.494. So two things to say. Number one, here's a complete analysis then of the population. We know that just over 30% of the population is homozygous dominant. We know that 20% of the population is homozygous recessive, and just shy of 50% of our population is heterozygotes. Okay? The other thing to say is if you do this math out, you might come up with 0.493 or 0.495 or something like that. And when you add it up, it might be just slightly more or less than 1. But that is just some rounding issues that you will encounter every now and again. Not a big deal. Okay, so that's a Hardy-Weinberg analysis. And you should be able to apply that to all kinds of population. Key first step, right? Key first step right here. Focus in on the recessive population because that is a guaranteed way to establish what Q squared is all about. Okay, take care. Hope that was helpful.